Hello guys, welcome to Lifeline again. This week we will be talking about advanced genetics. As compared to last week where we saw basic genetics or Mendelian genetics, this time we're going to get into the less common types of inheritance. This week is the second to last week, so we're almost there. And you will be getting 3 PPP points this week when you do your quiz, of course. A couple of reminders, as I was telling you, the week 14 PPP quiz is going to be due as usual on Saturday at 11:59 p.m. In addition to that, we have the week 14 quiz. This one is going to be due on your specific day of Lifeline next week. For you, this means that you are going to be doing the quiz within those 50 minutes that we usually met for class. So that is going to be next week on Tuesday. Depending if you're in the 6 p.m. class, then you will have to come in and complete your quiz from 6 p.m. to 6:50 p.m. Or if you are in the 7 p.m. class, you will complete your quiz from 7 p.m. to 7:50 p.m. You have 15 minutes to complete this quiz, and it is only 10 questions, so it is more than enough time to finish it. As usual, again, you only need to get 70% and above in order to get the full point. Fact or Fiction Group 8 is finally due on April 18th, and that's the last time we have Fact or Fiction this semester. Lastly, SPOTS evaluations will open on April 18th and are due by April 24th. Now, it is yet to be determined if we're going to give points for you completing the SPOTS evaluation. If we are going to give you points, then I will let you know in an announcement so that you can send me a screenshot or upload a screenshot of your completion so that I can give you that point. And if we don't have points this semester for it, then I would still greatly appreciate any feedback you have for me. So let's get into it. First of all, there is complete dominance. This is the most relatable kind. This is the one that we've already have a looked at without even naming it before. Complete dominance are just examples of what we did last week. In complete dominance, if the genotype of the offspring is heterozygous, then it will have the same phenotype as the homozygous dominant parent. So last week we talked about masking, alright? Masking meaning that the dominant allele is able to completely overrule the recessive allele and show that phenotype. As you can see here, the allele for brown is completely overruling the allele for blue, and the eyes look just as brown as they do in the heterozygous parent, right here. The example is if brown eyes is the dominant allele, it will mask the recessive allele for blue in the offspring with the heterozygous. Now, in order to express the recessive allele, you have got to have both recessive alleles. Incomplete dominance is the first kind that we have not discussed yet, but similarly, it only has to do with two alleles. The heterozygote offspring shows a different phenotype than either homozygous parent, so what we are saying here is that we're coming in with a new phenotype, something that neither of the parents showed, unless the parent was a heterozygote. So let's have a look at it. The offspring's phenotype is a blend of the parent's phenotype, which is why it does not look like either parent. No dominant or recessive allele. They are both equal, if you will. In this flower, red and white are neither dominant nor recessive, so their heterozygous offspring will not fully display either phenotype. The genotype big R is red, but it's not the same as the big R, as it was in complete dominance. Remember, just in the previous slide, we have a big B, brown eye. Big B, is still brown eye. Here, we have a big R, red flower, little r, white flower, but a big R and a little r, pink, which is different from big R. This is the definition of incomplete dominance, where you can almost say that both alleles, the big R and the little r, they have the same pull. So there is no masking effect here. They mix each other. In codominance, alleles do not exhibit complete dominance. Similar as in the last slide, there is no dominant or recessive allele here. So they are both fully expressed. Neither allele is dominant nor recessive. The phenotype of the heterozygotes will express the phenotype of both homozygous parents. So now, the heterozygote is not going to be totally different from either parent. This time, it's actually going to be equal amounts of both. Can distinguish between the two alleles. Phenotypes are not blended. So here you have it. This is the example where a pure red cow, or bull, if you will, has the genotype of big R. A full pure white cow has the WW, and you can see that the heterozygote is going to be a mixture of both of them. It is not blended as it was in incomplete dominance, but you can definitely see both genotypes being shared. So let's see if you get the differences. Which is an example of complete dominance, which one is incomplete dominance, and which one is codominance. So this first example here is showing complete dominance, right? Because we can see that the heterozygote is exactly the same as the homozygous dominant, and there is nothing of the homozygous recessive here. This example here, this flower compared to this flower, is expressing codominance because you can clearly see both the white and the red in this flower. This one, on the other hand, is showing incomplete dominance because it is clearly blended. The white with the red has blended in this offspring. So we can guess that both of these are heterozygous. Multiple alleles. One gene can have 3 or more possible alleles. So far, we've only been talking about the possibility of 2 alleles. For instance, whenever we mention the example of the eyes, we always say color brown, color blue, color brown, color blue. But clearly, there is more than one eye color in the world, isn't there? There are green eyes, there are blue eyes, there are brown eyes, hazel, right? So, what is going on here? The thing is that humans only have one pair of each chromosome, and therefore, they can only have up to 2 different alleles, even though there are plenty more out there. So even though there are alleles for green eyes, and brown eyes, and hazel eyes, and blue eyes, we as humans are only capable of having 2 of those. In the case of the example, brown and blue, we're talking about a human who happens to have only those 2 alleles. It has 1 allele for brown, 1 allele for blue, and that is because likely their parents did not even have an allele for green in them. That is how this works. There is plenty of those out there. While it is possible for a gene to have multiple alleles, a person can only have 2 because of the reasons that we just described. The example that we are most concerned with here is the ABO blood group system. Let's get into that. Blood components. And there are many more components to blood, but what we are concerned with here are going to be 3 things. The erythrocytes, which are the red blood cells, the antigens on the erythrocytes, and the antibodies on the plasma of the blood. So antigens are modified glycoproteins on the plasma membrane of red blood cells that stimulate the production of antibodies. Then antibodies are modified proteins produced by white blood cells that are created to neutralize foreign objects by attaching to a specific antigen. Now, this is the important part, foreign objects, alright? we are not going to get too much into the immune or the adaptive immune response that the body mounts against a foreign substance. But here is what you need to understand. Our immune system is built to react to anything that is not ours. Anything foreign promotes the buildup of antibodies. So let's have a look at it. The blood groups. Phenotype A can have two genotypes. Here, we're talking about complete dominance. In genotype A, the recessive allele O can be totally masked by the presence of the dominant allele A. In the phenotype B, we're still talking about codominance because the homozygous dominant shows the antigen B, and if the genotype is BO, it's still just the antigen B. Now, the phenotype AB is an example of codominance because A and B are they have equal pools, if you will, again. Equal pools means that the erythrocyte is going to show both antigens, as you can see here. Antigens A plus B, it has both of them. This one was an example of complete dominance, both of these were. Now this one is an example of codominance because both of them are being equally expressed. Phenotype O has only one genotype. Now this is considered complete dominance again, because in order to express that phenotype, you need to have both recessive alleles, and that is OO. The OO genotype has no antigens on it. Now let me just clarify this. There are plenty more antigens than A and B in erythrocytes, okay? What we say by no antigens, it means that it has neither A or B, as it says here. Alright? Neither A or B. And let's just make it clear that the O is a recessive allele. Before we move from this slide, I just want you to notice this trend. A person who has the antigen A on the red blood cells will not produce antibodies for A, because that would mean that it's attacking itself. It will become like an autoimmune disease. So, what are they able to produce? They are able to produce antibody for B. Now, a person with the B antigen on the blood cells is not going to produce antibody for B. It will produce antibody for A. A person with both A and B is not going to produce any antibody for these antigens because it has both and it does not want to attack itself, right? Now, a person without antigens, without A or B antigens, is going to produce both antibodies for A and antibodies for B. Now, it is this production of antibodies versus antigens that determines what type of blood a human is able to get. And it is all based on that immune response. We do not want to give someone blood that is going to cause their immune system to produce antibodies against that blood. Because if that happens, then likely the blood transfusion is going to do more harm than good. Let's continue with the blood groups. An individual will produce antigens for the blood type they have. So if someone is blood type AB, that means that they have antigens AB in their blood cells, and antibodies for the other one that they don't have. Someone with AB, again, will not produce antibodies for any of those because it has both. Blood type O produces no antigens but has both A and B antibodies. No antigens make it a universal donor. So, here is the thing. And I want to be totally honest with you. I, not until recently, totally understood how blood transfusions work. Because so far what we have said is that because blood type O has no antigens, pretty much anyone can get blood type O. But I kept thinking, but if I have blood type O, for example, and I want to give my blood to someone who is AB, am I not giving them antibodies with it? Am I not giving them antibodies A and B that I already have in my blood? Isn't that going to affect them? And hold that thought, because I will give you the answer as soon as I explain how blood donation works. Blood type AB produces antigens for A and B, but no antibodies. So no antibodies in that blood type means that the AB blood type is a universal recipient. It can get blood from anyone. Let's look at this. This is a specific for Keating students, all right, for Dr. Keating students. Instead of AO, it can be expressed as AI. All right, the I represents the O, basically. That's it. For Dr. Brooks students, you can stay with the AO. BO is the same as BI. I is the recessive allele. AB is still the same, and OO is II. So basically, if you're a Dr. Keating student, you want to substitute the O for I. That's it. No more than that. So here is the blood donation chart. Let's see if we get it, and I'll explain where my confusion was and how I got to understand it. So, the recipient is on this side. The blood donor is on this side. So, a recipient with blood type O, what antigens do they have? If you said none, you're correct. They don't have antigens on their blood cells. And again, please, there are antigens there. What I mean is that neither A or B antigens are present on the erythrocytes. So there you have it. It has neither A or B. So it is able to receive blood from O because it has neither antigens. Will a recipient with blood type O be able to receive blood from a donor who's A, who has antigens A in their blood? No, they won't be able to. What about B? They won't be able to either, because they, as a O recipient, are going to build antibodies against A and B. Therefore, A and B is also out of the question. Now, if a recipient A is going to receive blood, can they receive blood from O? Yes, they can, because O have no antigens. So, the recipient is unable to make any antibodies that are going to harm the blood cells that are being transfused into them. But here was my confusion before. Okay? As a blood donor who is O, you do not have antigens A and B. But you have antibodies A and B, don't you? That's the thing. Okay? You do not build these antibodies until you are exposed to the element. This is a function of the adaptive immune system, alright? And this is probably slightly out of scope for you guys, but I don't want you to have that doubt as I have for so long. The other day, I talked about this with one of the doctors who I work with in the ER, and they explained to me that if I'm a person who has blood type O, I do not have antibodies for A and B in my blood yet, unless I have been exposed to blood of the other person, of the other blood type. Alright? Which is why a person who is blood type O and has never had an infusion before will be able to donate pretty much to anyone, so long as they do not have those antibodies in their blood. Alright? Alright. So, now, will a person from who is recipient A be able to get some blood from an A donor? Yes, they will. What about B? No, because A makes antibodies for B. And as soon as those erythrocytes with the B antigen go into the bloodstream, recipient A will begin making antibodies for B and attack itself. So A and B is also out of the question because it also has B antibodies. What about B? Can it get from O? Yes, it can. So long as there are no antibodies there, O erythrocytes have no antigens in there. So the antibody A that the recipient B produces is not going to affect it. If you give them blood A, however, they will reject it because a recipient B will make antibodies for A. But they can get from B, and they cannot get from AB because they have the antigen A. What about AB? We already said AB is the universal recipient, so it can get from anybody. O, why can they get from anybody? Because they will not make any type of antibody A or B. So without the antibodies, they can get pretty much any type of blood. The Rh factor. Now, this is where you usually hear A negative or B negative or O negative type of blood. Rh is another type of antigen, alright? A type of protein on the surface on the red blood cells. Rh negative blood cells lack the Rh antigen, so they are recessive. This is an example of what? Complete dominance, incomplete dominance, or codominance. This is going to be complete dominance, guys. So Rh positive individuals can be either big R, or big R, little r. So the big R allele here is dominant, and this is an expression of complete dominance. Is the Rh, and there you have it. It is complete dominance. Erythroblastosis fetalis. Now, this is a specific example of why the Rh factor is important. Erythroblastosis fetalis is Rh incompatibility during pregnancy. A woman is at risk when she is Rh negative and her partner is Rh positive. Let's understand why. The first pregnancy will have no issues, but during birth, when the maternal and fetal blood mixes, Rh antibodies are made. So, let's talk about this. Remember when I told you that my big confusion was because I thought that a person with O blood already has antibodies A and B in their blood, as it is. That is not true. That is exactly the same reason why a woman in the first pregnancy would have no issues. Think about this. If the man in the relationship is Rh positive and the woman is Rh negative, chances are, actually not just chances, any type of kid they have is going to be Rh positive. Anytime. If the male is Rh positive, that kid will be Rh positive by default, because this is complete dominance, correct? So, in the first pregnancy, there are no issues because during gestation, the baby's or the fetus's bloodstream is separate from the mother's. But during birth, this blood mixes, and now that the Rh positive blood has mixed with the mother's Rh negative blood, guess what the mother is going to do? She will begin producing antibodies for Rh because she does not have Rh. Rh is foreign for the body, so antibodies will be made. Next time that this woman has a baby with the same father, for example, who is Rh positive, that baby will be Rh positive. But here is the problem now. In the first pregnancy, the mother was not exposed to the Rh positive, to the Rh factor until birth. So there was no issue there. But now that she has had a kid and the bloods have already mixed, guess what the mother already has in her system? Now she has anti-Rh antibodies. Now she has those, and whenever a new baby begins to develop with the Rh positive blood, the body is not going to like that, and it's going to attack the fetus because it will determine the fetus to be foreign, which it is, to be fair, but the Rh positive factor is what the body is targeting at that point. The baby has no option but to have the Rh factor, but the body of the woman already has antibodies for Rh, and that is going to cause an immune attack on the new developing fetus. Thankfully, nowadays, this is something that we can fight. There is a medicine called Rogam that is given to all Rh negative pregnant women to prevent this from happening. Or blood transfusions are also an option for the baby in that case. This is what's happening. See, the Rh positive baby with the Rh negative mother during birth is going to mix blood, the Rh positive blood with the mother. After that, the mother will develop anti-Rh antibodies, and when a new baby comes along who is positive, These antibodies are already present and will not allow the baby to develop further because of the immune attack on the baby's blood. Polygenic traits. These traits require the interaction of two or more genes. This is basically saying that this feature in a person, like skin color, has many genes at play, determining the type of skin color that one is going to have. Other examples include hair color, eye color. Sex-linked genes. These are important, guys. These are the types of problems that you will be asked to solve in your exam. Sex linkage problems. They are important, and let's see why. Some genes are only located in the sex chromosome. The sex chromosome, remember, in a woman is always XX, and in the man is XY. Females receive an X from their mother and an X from their father. Always. The father only has an X or a Y to give. The mother has two X's to give. It can give either one of them to the gamete in question. Males receive an X chromosome from their mother and a Y chromosome from their father. It is because they received that Y chromosome from the father that they became males. And the only thing they can get from the mother is an X chromosome. Sex-linked inheritance. Most sex chromosomal genes are on the X chromosome, so sex-linked traits do not follow standard Mendelian inheritance patterns. They do not. By far they don't. Because there is a discrepancy. There is a big difference between a man who only has one X chromosome and a female who has two X chromosomes. The way that we look at this is that the females pretty much have like a spare. They have the chance to be heterozygous, and therefore they have a chance to not show the mutant allele. Whereas males only have that one X chromosome. The Y chromosome is relatively small, so there is not as much information in that chromosome. What has the more information is the X chromosome, and whatever is on that X chromosome for that male is by default what's going to be expressed. Example of this is color blindness. The only major sex-linked gene on the Y chromosome is the SRY gene, which is the male determining gene. That gene has it transcribes for the production of several different proteins that aid in the conversion of a male during development. But the X chromosome can have several other genes that show sex linkage patterns. The example here is color blindness. There's another example like hemophilia, which is only on the X chromosome. So heterozygous females can have the gene but not express the disease phenotype because they carry two forms of the gene. So this is an example of complete dominance. See, this example is about hemophilia. A father who does not have hemophilia has to be with the dominant allele on the X and the Y because they have to have the Y if they're a male. Whereas the female can be a carrier, meaning that she can have the allele for the disease without herself having the disease because the other gene does not have the disease. Okay? And then we're gonna look at the offspring later in a better, easier way to see in a Punnett square. One mutant and one normal allele. They are carriers as they can pass it down to their offspring. So even though that female may not have the disease herself, she can have the allele within her, and she has the capability of giving it to one of her offspring. Because males have only one X chromosome, they can only show the trait or not show the trait. That's all we can do. There is a special name for this. For males, in instances like these, with hemophilia, males are hemizygous, meaning that they can only show what they are given by the mother. That's it. There is no backup. So that is what hemizygous means. While females can be heterozygous. Most X-linked recessive diseases are found in males due to having only one X chromosome. So there is a huge discrepancy in how this works. By far less ladies than males have the disease in question. X-linked recessive combinations. There is the homozygous dominant, where the disease is not present, and by default this would have to be a female, right, in order to be homozygous dominant. Heterozygous, a carrier, has to be a female, meaning that she can have the gene, the mutant gene, without herself having the disease. Homozygous recessive, when a female actually has both recessive alleles and the disease is present and not present. This is the example of the genotypes. We express it with an X, and then we put the letters as a subscript, as an upper script for this. See, this is the heterozygote with the lowercase a, but still X. This determines that this is sex linkage. Now this is the case for males who have a Y by default. If they have the recessive one, they have the disease. If they have the dominant, they do not have the disease. That's simple. They are hemizygous. Color blindness. Red-green color blindness is a recessive X-linked trait. Males have only one X chromosome, which comes from the mother. Females have two X chromosomes, one from the mother and one from the father. Need to inherit trait from both parents to express, if you are a female. Men with the red-green color blindness inherited, that gene came from their mothers. That's the only place where it could have come from, the mother. Because if they are male, what did the father give them? The Y chromosome. Men receive the Y chromosome from their fathers, which cannot carry that gene. So this is color blindness. It's basically in the retina. This is a little bit out of scope again. The retina has 3 types of cones, basically. These cones can detect either green, red, or blue light. The cones for red and green have been affected in this color of disease, in this type of disease. So blue is somehow more seen than the red and green colors and the mixtures of them. And this is what we're looking at, basically. A carrier mother who has the mutant gene but the normal allele, and an unaffected father who has only the normal allele and the Y chromosome. When they have kids, these are the chances. The mother always has to give an X. It can give either the bad X or the good X. Alright? The mutant X or the normal X. The father can only give an X or a Y. When the mother gives the mutant one, here in these examples, and the father gives the other X, the daughters are going to have these genotypes, these phenotypes. An unaffected daughter, because the father can only give that X, that good X, and the mother can give either the bad X or the good X. You can have a carrier daughter. However, when we talk about the sons, the father can only give the Y allele, the Y chromosome, sorry. So it's up to the mother which gene they get. Do they get the mutant one, in which case the son will be affected, or they get the dominant allele, in which case the son will be normal. In the case where the mother is unaffected and the father is affected, pretty much any daughter is going to be a carrier, because in order to have a daughter, the male has to give that one X chromosome. Alright? So both daughters possible are going to have the gene, but not express it because the mother has no recessive allele to show. They are going to be carriers, whereas the sons are going to be by default unaffected and not a carrier because they can't be. They were given the Y chromosome, and then they were given the only type of chromosome that the mother has, the normal chromosome. Now, let's do a review to see if you understood this. What kind of dominance is presented in the picture above? So we have a red and a white flower producing a pink flower. This is called incomplete dominance. What about here? We have a blue bird with a white bird producing a blue and a white bird. This is an example of codominance. Review question. So, if a color-blind man marries a woman with normal vision but is a carrier, what percentage of their sons would be colorblind? This is an example where you have got to do a Punnett square. Let's see how we would go about it. As always, begin by getting the possible gametes from the father and the mother. Let's look at the woman first. A woman with normal vision but is a carrier. This means that she has got to have one X with the normal allele and one X with the mutant allele. There you have it. That's the mother. Two X's, one big, one small. The father says that he has normal vision. By default, the one X that father has got to be a normal X. Sorry, a color-blind man. Okay? Not normal vision. A color-blind man would have to have by default that recessive allele. And the other chromosome has got to be a Y. Now fill out the Punnett square as you've been shown how to do. X with big C, X with little c. This daughter is a carrier, does not have the disease. This daughter, X little c, she is a colorblind girl. This son right here, and remember, in this type of problem, it matters if we're asking about the sons or the daughters, because there are two types. They can be daughters or they can be sons. One son is going to be normal. The other one is going to have the mutant allele that came from the mother and the Y that came from the father. So what percentage is going to be colorblind? 50% of the sons will be. It is important to recognize that it matters if they are asking about sons or daughters or offspring as a whole. Because if the father was unaffected, this would be a capital C. Alright? Let's illustrate it. So this would be a big C, and therefore, this girl would have never been a carrier, and this girl would have only been a carrier. So, no daughter would have had the disease in this question. So, if the question says, how many of the offspring would show color blindness, the answer here would change to 25%. However, if the question is still asking how many of the sons would be colorblind, the answer is still 50% because out of the two types of sons, one is colorblind. The other one is not. So 50% is the right answer. Another question. If a colorblind man marries a homozygous woman with normal vision, what percentage of their sons would be colorblind? Let's see. Homozygous woman, both big C's. Colorblind man, as you can see there. Fill out the Punnett square. What percentage of their sons would be colorblind? None of them would be, because the woman has no capability of giving an X with the mutant allele. However, both daughters would be carriers. Now, let's review. This is from last week. This is a dihybrid cross, and we're going to fill it out to see that you understand how to do these for your exam. So, in a flowering plant, tall, big T is dominant for short, little t, and blue flowers, big B is dominant to white flowers, little b. A tall plant with white flowers, there you have your genotype, is crossed with a short plant with blue flowers. There's your genotype. What is the chance that the offspring will be short with white flowers. And these are your options. You will see that it is quite difficult to find an answer without actually doing the whole square. So we're going to go ahead and do it. First, you have to FOIL, right? So we need to get your gametes. First, the big T with the little b. There you have one. Little t, little b. There you have it. Then another T, big T, with a little b. That's this one. And then another little t with a little b. There you have it. Now with this one, little t, big B. There you have it. Little t, little b. There you have it. Little t, big B. Again, little t, little b. There you have it. Now, what is the chance that the offspring will show white will be short with white flowers? Fill the square out, and let's watch out for when we get short. This is the genotype that we're looking for. Short with white flowers. This is the genotype we're looking for. There's one. There's another one. There's another one. And there's another one. So we have 4 out of 16. Where's the answer? Nowhere to be found, apparently. But remember, simple math tells us that 4 out of 16 is the same as 1 out of 4. There's your answer. E is the answer. Okay? Now, this was basically it, guys. This was just a review from what we did last week, among a bunch of new things that we have just explained to you. So please, keep an eye out for this. The discussion board will be open as usual. If you have any questions, go ahead and ask me. If you'd rather ask me in an email, go for it. I will be happy to answer it for you. And with that being said, please do not forget to do your PPP quiz so that you can get points. This one is especially big. This one is 3 points. Do not miss them. Do the quiz by the deadline on Saturday at 11:59 p.m., please. Stay safe and have a good rest of your day. Bye-bye.